action potential of a neuron. First of all, an action potential is the change in electrical potential associated with the passage of an impulse along the membrane of either a muscle or a nerve cell. In this case, it's a nerve cell. The action potential is propagated into one direction down the axon. So it will go from the cell body down to the axon and eventually to the terminal knobs. And the action potential is seen in red. It's going to go from the left to the right. As it occurs, the inside of the axon becomes less negative or closer to zero. This is called depolarization. It occurs when sodium, which is usually on the outside of the cell, enters the membrane. When depolarization reaches threshold, then action potential occurs next. So it will go from the left to the right. The recently depolarized area of the membrane is in refractory period, which explains why action potential is in one direction. As I explained before, sodium ions are on the inside while potassium is on the outside of the neuron. As action potential occurs, the sodium gates open and sodium rush into the cell, causing depolarization. The change of the inside gets closer to zero or less negative. Next, sodium channels close and potassium gates open. When potassium gates open, the cell repolarizes, which means the inside is negative and the outside is positive, which is back to normal. The last thing to occur is the sodium-potassium pump. What it does is it helps restore the resting potential in the membrane. So the potassium will go outside and the sodium goes back inside. Finally, the action potential goes to the presynaptic terminal, also known as the synaptic knobs, and causes the calcium channels to open, as seen in the blue. Calcium then enters into the terminal knobs through the calcium channels. The calcium causes the synaptic vessels to release a neurotransmitter called acetylcholine, which is inside the vesicles. This is a close-up of the synapse between the terminal knobs of one neuron and the dendrites of another. The acetylcholine molecules bind to the receptor sites on the sodium channels of the dendrites of the next neuron. Because the acetylcholine binds to the receptors, it causes the sodium gates to open, causing sodium ions to rush into the next cell. This action makes the membrane of the next neuron less negative or close to zero. If the membrane reaches the threshold, then the action potential will be created for the next neuron.